Now, what if there is oxygen? If there is oxygen, this 3-carbon molecule pyruvate, these guys will go on to enter the mitochondria. Remember the mitochondria? It's an organelle inside of the cell, so we're still inside of the same cell. And the mitochondria has a double membrane, right? So membrane number 1, membrane number 2. So here, right here is inside of the mitochondria. And so pyruvate, if oxygen is present, so if yes, oxygen, then pyruvate will go into the cell.
And in one of your videos, we actually talk about how it goes into the mitochondria through a series of channels.
And it is in the mitochondria that the citric acid cycle, I should say the citric acid cycle, which is why a lot of people call it the TCA cycle. And then other people will also call this the Krebs cycle, named after the guy who came up with this or who discovered this. Ain't it always? All right. So either way, you call it is fine, as long as you know they're talking about the same thing. This is what happens in the mitochondria if oxygen is present, all right? And the cycle occurs 1 pyruvate at a time. So we're actually going to just focus on as 1 pyruvate at a time, and then we'll do math later. Sound good? All right, so we have pyruvate inside of the mitochondria. Yay! So the citric acid cycle is ready to go. Pyruvate, once it enters, it can go in 2 different directions, all right? Direction number 1 that it can go, and notice that this is a 1-direction arrow. This means that it's a non-reversible reaction. So one way that it can go is it can be converted into acetyl coenzyme A, and this is a 2-carbon molecule. So how does this happen? This happens by NAD plus working to pick up and convert into NADH, and CO2 is also lost in this reaction. And so that's why this molecule of pyruvate, which was a 3-carbon molecule, has gone down to a 2-carbon molecule, because one of those carbons was let go through the release of carbon dioxide. And this process is done by, you might have figured it out because you see NADH, right? This is done by a dehydrogenase enzyme. Specifically, and this name makes sense, pyruvate dehydrogenase. Do I have room? Well, that's good enough. All right, sound good? So this is where we have our first loss of carbon, 1 lost carbon. The other direction that pyruvate can go when it enters into the mitochondria is it can also go through another non-reversible reaction via pyruvate carboxylase, which converts pyruvate into something called oxaloacetate. This is really important for the cycle. This right here is a 4-carbon molecule, hence the name of our enzyme. It's called a carboxylase because it adds a carbon to our molecule, all right? So in one direction we're losing a carbon, in the other direction we're gaining carbon. Both are absolutely critical to the citric acid cycle. So let's keep it going. The way that the cycle goes is actually in this direction. But this step right here over to the left is very important because it actually contributes to step 2 or 3, depending on how you look at it, in the citric acid cycle. So let's break it down. We have our acetyl-CoA, which is a 2-carbon molecule. And the next step in the cycle is to actually convert acetyl-CoA into citrate. And citrate, hence the name citric acid cycle, citrate is a 6-carbon molecule. And to get acetyl-CoA to somehow magically become citrate, you need 4 more carbons, right? Where can we get those 4 carbons? Oh, I know, we can get it from over here, right? So that's how it comes into play. And just to make things super clear, pyruvate can't be both oxaloacetate and acetyl-CoA. The point of this diagram is to show you that pyruvates are constantly entering the mitochondria because you are eating glucose, and glucose is constantly going through the cycle and constantly entering mitochondria, right? So some pyruvate will go to acetyl-CoA, some will go to oxaloacetate, and them coming together is what helps produce citrate, our 6-carbon molecule, which is where our cycle gets its name, the citric acid cycle. Citrate, a lot of people also call citric acid, just as an FYI. So we have our 6-carbon molecule, and now we're entering the reversible phases. So the phases that are at equilibrium and are controlled by the concentration of each substrate, right? Substrate or product, depending on how you look at it. So citrate can then be converted into isocitrate, and this is via an enzyme called aconitase. Do I care that you know that? Not really. Just remember ACE means enzyme. It's doing things. Cool? All right, so this is still 6 carbons. It basically just rearranged the carbons. All right, rearranged the molecule a bit, but still 6. Next, we have a unidirectional reaction where isocitrate is converted into alpha-ketoglutarate. Alpha-ketoglutarate, though, is a 5-carbon molecule. And so what happens to get isocitrate to alpha-ketoglutarate is we have energy produced in the process. So NAD plus picks up a hydrogen, or a hydride, right, and which produces NADH, and we lose another carbon through CO2. So there's where we lost the carbon that was there. It was lost to CO2. So can you guess what enzyme probably did this, or what type of enzyme? If you had to choose between a kinase, a carboxylase, or a dehydrogenase, what likely is responsible? Right, this is a dehydrogenase enzyme, okay? Specifically, it's called isocitrate dehydrogenase, which makes sense because it specifically dehydrogenizes, if you want to call it that, isocitrate into alpha-ketoglutarate. So here, I'm just going to go ahead and put some jazz on the NADH to help us keep track of the fact that we have now produced energy, or a source of energy. And then over here, I'm also going to mark that we lost one more lost carbon, hence why we have 5 carbon molecule right here in alpha-ketoglutarate. All right, so we are now halfway through the cycle. You got this, guys. I believe in you. And hopefully this is kind of making sense that you can see some semblance of a pattern in how this works, all right? So the next step is alpha-ketoglutarate is then converted into succinyl-CoA. And so how does that happen? So succinyl-CoA is a 4-carbon molecule. A similar process as to what you just saw before happened. So let's see if you can fill in the gaps on your own now, right? What was likely put in to this reaction to produce succinyl-CoA from alpha-ketoglutarate? Right, we used NAD plus as a acceptor of that hydrogen, so that forms NADH, right? And we lost another carbon, so that means that CO2 is likely released, which it was, so that's where we lost that carbon from. Boop. So what kind of enzyme is probably responsible for this reaction? Right, another dehydrogenase, okay? Another dehydrogenase is responsible for this reaction, and the name of the dehydrogenase is alpha-ketoglutarate dehydrogenase. See how the names kind of make sense? All right, so I'll go ahead and mark this guy as another lost carbon, so one more lost carbon. So we're halfway through our cycle, time to finish out the clock. So from succinyl-CoA, we produce something called succinate, and that is via an enzyme that is called thiokinase. However, I just want you guys to know that it's a kinase. So if we're talking about a kinase, what are you thinking of off the top of your head immediately? Right, kinases deal with phosphate groups. So normally this is where I tell you we are dealing with ATP and ADP. However, this is special. This is dealing with GDP and GTP. However, don't let this confuse you. They're basically equivalents of one another, all right? One GTP molecule can easily be converted into an ATP molecule, so you can think about it that way if you want to. Just know that technically we're dealing with GDP and not ADP here. So in essence, the moral of the story here is, boom, energy is produced in this step thanks to that kinase, which means that a phosphate group was taken and added to that GDP to produce GTP. From succinate, we have another reversible reaction thanks to a dehydrogenase enzyme, specifically succinate dehydrogenase, where another type of energy producing molecule, or energy contributing molecule called FAD, is converted into FADH. And this also is a source of energy, so I'll go ahead and make him yay. All right, so getting that nice hydrogen ion, kind of like we do with NADH. And this produces a molecule called fumarate. Fumarate then undergoes a chemical reaction with an enzyme called fumarase, which is basically literally translated into an enzyme that deals with fumarate. So very creative naming, guys, right? So fumarate via an enzyme called fumarase is converted into malate. And if the concentration of malate is high enough, then that reaction can reverse and fumarase can convert malate into fumarate again. But anywho, fumarate converted into malate to continue the cycle. And then, this is where it gets interesting, we have the last stage in our cycle where malate can be reversibly converted into oxaloacetate via malate dehydrogenase. And so we're dealing with a dehydrogenase again. So just like before, that means we are dealing with NAD plus being converted into NADH. Boom, 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 boom. All right? So here is our whole cycle, all right? And we were just focusing on what happens if we hone in on one pyruvate molecule. So from one pyruvate molecule, this whole cycle occurs. So let's go ahead and count up what we have, all right? From one pyruvate molecule, we have some NADHs, some GTPs, and some FADHs, okay? So let's count them up, shall we? For NADHs, we have 1, 2, 3, and 4. And again, that's just from one pyruvate molecule. So we have 4 NADHs. And then for GTPs, boop, we have 1 of those. And FADHs, we have 1 of those. All right, so we're going to hold on to that for later. I'm going to go ahead and write it down from one pyruvate. So now this is where things get interesting, all right? All of these energy sources then go to something called the electron transport chain. We're moving on from the citric acid cycle to something called oxidative phosphorylation, which deals with the electron transport chain and chemiosmosis, all right? So where does that take place? Do you guys remember where the electron transport chain occurs? Right, it takes place in the mitochondrial membrane, right? All of these products here are going to travel, all of these energy products are going to travel here Here we have the electron transport chain, all right? So I'm gonna go ahead and draw my little dotted lines. These guys are all gonna go boop. So off to the electron transport chain we go.